Okay, back for our last topic, which is polygenic inheritance. There is much, much more to the patterns of inheritance, but this is the last topic we're going to cover. So some traits are governed by one allele. We just heard all about them, but some are governed by more than one allele. In fact, probably most, because human traits can be very complex. Remember the scenario from 10th grade bio? The gene for blue eyes is recessive, the one for brown eyes is dominant. You have to be homozygous recessive for the blue eyed gene to have blue eyes. If you have a brown eyed gene, you will always have brown eyes because brown is dominant and it will always express itself. And then some joker in the class would say, but I have green eyes and my mom has hazel eyes. And what about people with one brown eye and one blue eye? Well, the truth is, nothing in life is all that simple. There are actually at least 10 that we know of alleles that govern eye color. And look at that chart right there, all the variations of eye color. There are two major ones, and the rest are minor ones. And they interact with the alleles that govern skin color and hair color, meaning they will express themselves differently if you also have the allele for increased melanin in your skin or hair. If you have red hair, your blue eye gene may act completely differently. Another example, there have been over 700 alleles that are identified as contributing to height, and they all play together. And they interact with the environment. So nothing is ever as simple as it seems. And most of our diseases are not governed by classical Mendelian inheritance. They are governed by other things. Another analogy, single gene traits are like apples. If I'm hungry and I want an apple, I only have to eat an apple. It expresses itself as an apple in its current form all by itself. But if I want cake, especially if I want to make my own cake, I have to mix different ingredients together. You can't just have flour and that's cake. A cake is much more than that. It's flour, it's sugar, it's eggs, it's cocoa powder, it's butter. And then it's things like how you combine those ingredients and how long you bake them and what temperature the oven is. And then icing has its own recipe. So that is more like polygenic inheritance, where you have more than one ingredient to form a trait. Those ingredients have to interact with one another, just like yeast and sugar makes a cake, makes a bread rise, or something like that. They have reactions within that allele pairing. Um, so that's an analogy that just kind of explains it briefly. Complex polygenic disorders are neither purely dominant nor recessive. Each allele adds or detracts from the expression of a trait, just like the ingredients in a cake add something to the chemistry, the flavor, the texture of that finished product, or they balance something else out. Phenotype depends on the interaction with other genes or environmental triggers. We have things like modifier genes that decrease the expression of traits. Okay? So it's complex. There's a liability model or a threshold model. And what this says is that there are alleles or versions of a gene that create risk. Sometimes having some risk alleles is actually adaptive, but when you have more than a certain number, it gives you that disease. For example, Fragile X. So, Fragile X is found on the X chromosome. If you have, say, 5 risk alleles on that X chromosome, you will not have Fragile X. If you have more than 20, you're probably a carrier for Fragile X because some of that material will transfer to someone else. It might be enough to give them Fragile X. But you need over 200 of them to actually express the disorder known as Fragile X. And for people who are unfamiliar with what that is, it is a disorder that is present in childhood. It can cause a type of autism. Most of the time, it's moderate, not quite what we used to call Asperger's, and not quite the profound disorder used to be called autism, but moderate autism. Pointy chin, large ears, protruding ears. So you need a certain number of those risk alleles to have Fragile X. You need fewer of them to be a carrier for Fragile X. And fewer than that, and I do believe the number is under, it might be 5 to 20 copies. You will not, it makes no difference. So it's sort of like, you have this much of something and it's fine. You put a teaspoon of salt in a cake and it increases the flavor. You put a little more of that and maybe it changes things. But if you mistake your sugar for salt, that really changes the flavor and the texture of the finished product. So that's what the liability model or threshold is. And that's going to be a concept that maybe explains certain things like hypertension or glucose metabolism. It's the number of risk alleles. And when we get better at identifying these risk alleles, we can test for them. And this is one of the problems that you might see with these direct to consumer marketing companies like 23andMe that give you those health reports, is that maybe we haven't identified all of the alleles that are associated with risks for certain disorders. For example, we know that type 2 diabetes has a genetic component. We know that not everybody who has a family history of diabetes gets it, but some people do, and they're at greater risk than anybody else. Well, if you take one of those 23andMe tests and it tells you that they didn't identify any of these risk alleles, that doesn't mean that you don't have them. It just means that 23andMe didn't find them because either they haven't been identified on the genome or 23andMe is not the best test in the world. So that's just a little information for you. Genograms and pedigrees a lot of times are very useful at hinting at the severity of genetic risk because they show you how many family members are affected. So that could tell you how many risk alleles are kind of circulating in your family tree. The last topic we're going to discuss is chromosomal disorders because they are polygenic to the max. This affects a whole chromosome or even a piece of a chromosome, but it's a mistake. Most of the time it occurs during meiosis 2 when we're getting ready to package that material for combination with somebody else's chromosomal information. Most often it affects the ovum because unlike men who are producing gametes all the time throughout their lifespan, women are born with all the eggs they're ever going to have. Now, going from meiosis 1 to meiosis 2, as that material gets older and the quality of the egg diminishes, we have more of a chance of making a mistake. Things can get sticky. So the chromosome, instead of splitting in half and giving you nice, gene separation, they kind of stick together. Now we have a trisomy, 3 copies of a chromosome instead of 2. So we are now 47 XX or XY. We have 47 chromosomes. One of them does not belong. Most of these are lethal. We'll talk about the exceptions on our very last slide. And that's not meant to confuse you, but most are lethal. 3 chromosomal disorders that have been identified that do result in the birth of a child who might live for a little while are 13, 18, and 21. 21 is known as Down syndrome, and that is the only that's the only trisomy when the person is a pure trisomy that is associated with living to adulthood. Trisomies 13 and 18 almost always result in either a miscarriage or a stillbirth or death in infancy. Most of them don't make it past the first year. There are isolated case reports of people living longer, and we'll talk about that maybe when we talk about mosaicism. But let's kind of get through. This is Patau syndrome. It's trisomy 13, and you can see the 3 copies. If the chromosome that's affected, let's say it's number 4 or it's number 11 or it's number 15, is a trisomy. Usually that will end in a miscarriage very, very early because as the proteins are being coded by the genes, things aren't coding right. You're not making the normal cells, the normal tissues, the normal organs. It's incompatible with life. But 13 happens to be one of the ones, the information that is duplicated is not as critical. Same thing with 18 and 21. It's pretty darn critical because 13 is not a good prognosis. But here we are with a monosomia. This is one chromosome. Actually, it should not say chromatid. It is a chromosome without a partner. So what we have here is 45X. 45 chromosomes. There is no X or Y chromosome to pair with that sex chromosome. Again, the information on chromosome X is maybe not as critical, or on that second sex chromosome is not as critical to life, but it is still pretty devastating to the individual who has it. It impacts height, it impacts reproductive characteristics, can lead to learning disabilities. So that's a monosomia. It's one of the only monosomies that does result in surviving offspring. Mosaicism, and this is our last slide, so congratulations, we got here. Mosaicism occurs when only some cells have that mistake. So here you see an individual. There's a normal cell, karyotype 46. I believe that's an XX. And then here, other cells are missing that second X chromosome. So we have 45X. So some cells code for Turner syndrome and some code for a normal phenotype. Now, depending on how many cells are affected and which tissues, organs, proteins are involved, the person can actually be phenotypically pretty normal. But when you examine their cells under a microscope with that staining, this is the karyotype that you're going to see in some of them. Most of the time, mom donates the normal 23 chromosomes, and dad donates the normal 23 chromosomes. They unite, forming 23 pair, 46 total. And it happens very early in embryonic life. Depending on when it happens exactly, determines the number and location of the cells. So we know that in early embryonic life, everything starts out as a stem cell, and then it sort of differentiates a little. We have the ectoderm, the mesoderm, and then those cells can migrate. And that's why you see the affected cells in certain areas of the body. It kind of depends on where that mistake happened in the chain of events. The earlier it happens, the more cells are affected. Now, I'm going to give you one last example before I cut this video off. And that is an example. It's actually, I can't remember if I found it in your textbook or if I found it while I was going down one of my many, many rabbit holes. There was a set of twins, identical twins, and mom had conceived via IVF. I don't think there was any pre-implantation diagnosis done. But sometimes in the IVF process, cells that are eukaryotic, they have a normal karyotype, can be damaged. Now, twin A was born and lived an hour. All of the cells that were examined had trisomy 18, which, like we said, is usually lethal. Twin B, on the other hand, was born and appeared perfectly normal, did not have rocker bottom feet, had all organs, everything intact. She grew up, she went to college, she was an athlete, had no known health disease, no problems. But when they looked at her karyotype, studying her serum and her cheek cells, they found this trisomy 18. So by all accounts, she should have been very, very disabled if alive. But in fact, her phenotype was normal. And so what they're saying is, in this account that I read of this set of twins, was that we can only study certain cells. Like, you're not going to do a brain biopsy and risk damaging healthy tissue to look and see if the brain cells are affected. You're not necessarily going to do a liver biopsy or muscle biopsy or any other tissue to see if the cells have trisomy 18 versus a normal karyotype. If the individual is healthy, you're not going to cause that risk to the individual. You're going to study the cells that are accessible, and you're going to be as least invasive as possible. Now, in the twin that died, they were able to do an autopsy and take sections of many different types of tissue and study, and they did find trisomy 18 in anything they found. But in this individual, it was decided by experts that the most likely explanation was that this person was mosaic and that the only cells that coded with the abnormal karyotype for this twin were the ones that they were testing in the serum and in the squamous epithelial cells inside the mouth. So that's what mosaicism is. That may become important when we discuss what pregenetic diagnosis can and can't do and the decision-making process and the ethics involved in that. Okay, and I think that's quite enough material. Go through it. You have a readiness assessment test for this material due Sunday night, 23:59 on September 11th. And, this information will be on your exam. I will keep giving you guidance and reminders when it comes to testable material because I know this content isn't always accessible. But let me know if you have any questions, and I will certainly try to answer them. Thanks, and good night.